Okay, so I'll get started today. We actually have quite a short session for our first session. We're just going to finish off RNAseq and do something which I think has become more interesting over the last few years. It's something it's hard to do in single cell, so it's still a lot of doing this in bulk. So where are we? We're on our page, rockefelleruniversity.github.io. I got to update the videos. I'm behind. I will do that today. We've done intro to R, plotting in R, reproducible R, so we should know how to do our work in R. We learned a little bit about genomics data and how we can look at that in IGV and how we can handle it using the Bioconductor libraries. The real point of all that was to allow us to start to do some actual analysis of high throughput sequencing data. And hopefully all that together, we should have a good understanding of how we can go from FASTQ with a reference genome, either from internal packages to Bioconductor, like the BS genome packages, to how we can produce effectively a quantification of gene, exon, or transcript. And then we went forward really with mostly the gene expression, quantification over the gene. And we looked a little bit at gene expression. We did a little bit of quantification of transcript, but we were summarizing it to gene level. And then we did differential gene expression. We did a little bit of visualization across groups. And then we looked at how we can use things like gene ontology and MSigDB to do some really simple enrichment analysis. So given we have a set of genes which go up, what functions do they contain? For the last session now, we're going to actually go back to exons and transcripts. So we'll try and cover the idea that really gene expression is great, but you can get points where a gene's expression doesn't change, but the transcripts within it do. And that can be an entire obvious alteration of the function of the gene. You could suddenly be expressing a transcript which does something very different to the original gene's function. And that could really be something very informative about what's going on. So, you know, if you ever look at development across time in certainly mouse and human, especially neuronal development, there's a lot of splicing which goes on to change the functions of these genes. So I think this is kind of a nice one. We're going to take some work. Actually, I worked on when I was back in London with this guy, Jesus Gill, who's a very big name in senescence. So, you know, the parts before you start doing death of a cell, sometimes a cell will dive into senescence. And this is very interesting for cancer biology. He himself, I think, is more interested just in the mechanisms of it. He does do some cancer biology. But we wrote this up a few years ago, and it got published in somewhere. We can find all the data on GEO. And this is all RNA-seq data. You know, plenty of replicates. And we have a knockout for, not a knockout, an SH, so a silencing. We're down-regulating it with an siRNA for PTB1. So PTB1 is a splicing factor, but it's not an essential splicing factor. I mean, you may never develop properly if you don't have it, but it's not part of the core machinery. So by silencing this, knocking this out, we should induce a particular program of splicing changes. Okay? PTB1 effectively guides the splicing of genes. So I've already done some of the work here, but if you went back to the original data, and remember on GEO, it's a little trickier to find, but perhaps we go to find the data here. We can pull out the SRA number. We can go to SRA. And from here, we can start to look at how we download the data. Personally, I prefer just to get this ID. And we go to, not necessarily grabbing food, but we'll go to ENA, European Nucleotide Archive. And we can always search our data there. It's going to be down, or no, we're up. Search session. It's a little slow. If we went back to, say, the study, you know, if I'm following on that GEO link, we should get here. Eventually, it will load. We have all the FASTQs we would need to actually start processing this data right from scratch. So if you want to, I invite you to do this. It's a good exercise following the tools we have learned in class, and that's exactly what I've done. I've aligned it with our subread using subjunc, and then I've summarized accounts in exons using summarized overlaps. And I've also done the transcript expression for this using salmon. So all gene and exon counts I've already done. It takes a little while, right? So we have that in the R data directory. You'll find, sorry, in the data directory, and you'll find data/senescence exon counts.r data if you want to follow along. So in our first session, we saw there are two separate ways to gain gene expression estimates from the raw FASTQ data. In our second session, we imported these and we actually saw how we could then capture gene expression and use this to identify differences in the conditions for particular genes. So this gene went up in one condition, this went down. So now we're going to concentrate not on necessarily gene expression changes, but differences in exon usage, and then how that may be relate to transcript. Okay. So we saw in session 2, we can use these BAM file lists, or BAM file. It's like an object which comes from the R SAMTools library. So once we've loaded that, we just give a vector of everything we want to count over. So these would be the full paths to your BAM files. We want them to be indexed 2. So we hope there would be a BAM.bai in there, and they appear to be sorted. So we've already sorted them here. Next thing we would want to do is pass the BAM file list to count. So we basically pass all the paths to the BAM files. We just need to tell it the yield size. And if you remember, this is going to be important for your laptop or for your HPC. This means we're reading in 10,000 from every BAM file as we try and process it. So 10,000 is quite small. You could probably do 1 million. If you're on a massive HPC, set that to 20 million. And that will just speed up your process. Here on your laptop, you could want to keep this to 1 million. So for differential exon usage, so I think previously when we did this, we were then counting over genes. So for differential exon usage, we need to actually create a new set of non-overlapping disjoint exons. And it's those that we will count over. I'm going to show you what those look like in a few slides. We looked at it very briefly previously, but effectively exons overlap, and we need to try and create a non-overlapping set of exons, disjoint such that they don't actually overlap. And it's from those we will do the counts. To produce those, it used to be a real big pain. So DexSeq, the software, so I think we're on DexSeq 2 now, but DexSeq, the original software, used to rely on you installing Python and then running this Python script, DexSeq prepare annotation. That only really worked on one particular annotation type. I think it worked on GenCode, and it didn't really work on anything else. There was something wrong there, right? We're passing from R to a separate Python script and asking. I mean, even if Python does wonderful things, we don't necessarily need to call it here. People like me started building our own functions in R to do this. We don't need to anymore. We've got this wonderful function available to us, disjoint exons. I think I said this last time. This is a new function, and they've decided to change its name. So I'm teaching it this year. Next year, I will have to change the name of the function. And we'll probably start using this function, exonic paths. If you ever see this, warning, this is deprecated, you're okay for now. It's going to work. It's only a warning. It's telling you, if you're teaching this course, update your slides next year. But it's also saying, you know, you guys be aware, if you're writing your own code, you're writing your own software, start using exonic paths. Okay? So it can sometimes take 1 year, it can sometimes take 2 years. We see a lot of this in the tidyverse packages, dplyr, ggplot. They move very fast. So we just need to supply to our disjoint exons function, which comes from the genomic features package, our txdb object we want to extract the exons from. So we don't need to run anything like genes or exons by. We just provide the actual object, and then it's going to produce this granges object containing all the non-overlapping exons. a much better example, I feel like last time we had this, but anyway. So once we've got these G ranges, we then want to assign some names to them. And what we're going to do is effectively take the gene ID from the mcols, the metadata column. So you can actually see that here. And we're going to concatenate that with the exon part, which just tells us the position that was this exon the first exon, second exon. And that just gives us a unique ID here, which contains gene name, Entree's gene ID here, and then the exon part. Okay. So something unique we can trace back to the original G ranges. So the reason we want to do disjoint exons, so if we look here, we can see this is the actual ref gene annotation for, I can't quite read that, HSH2D. And it looks complex, right? We have 1, 2, 3, 4, 5, 6 different transcripts, and they don't differ in that much. It looks like the first exons are all the same. Oh, sorry, the first exons are all the same. Here we have a distinct exon for the second transcript, and if you look over here, it's getting a little complex. We probably want to have a zoom in on that, and then maybe we have something here too. We zoom in on what was going on in that part there. You can see it is kind of complex. And what it's trying to do is basically pull out for us an exon part, which allows us to represent every exon there. So we have an intron retention here. So this is when we have one long exon over this, what would be an intron in all the other versions of the transcript. And so that means we keep this exon, we gain an exon here, which represents most of this. We seem to have a little bit of an exon sticking out for this transcript here. It's hard to tell at my angle. But if we want to represent that, we also need another exon part, and that represents any, I guess, signal which comes from either of these exons. And then finally, you have this exon. And then, if you look very carefully, this exon stops a little short, which means we have this exon. Okay, so we're not doing anything yet computational, but you could imagine if I saw an increase in expression over this middle exon, very simply, I would say this transcript or this transcript must be going up, right? If I see more signal over this intron, this exon here, this kind of this retained exon, because it could belong to no other transcripts, right? It needs to belong to something which had an intron, an exon retention over the intron. So now we have this disjoint exons. We can use the same trick that we did previously to summarize over genes. Okay, we just need to do it a little differently. Previously, when we summarized over genes, we provided a G ranges list. Okay, so the non-overlapping exons is just a G ranges. Effectively, I mean, you can think of this very simply. When you pass a G ranges list to summarize overlaps, it counts all the reads in every element of the list. So it counts all the reads in every exon belonging to a gene. Here we're going to pass a G ranges, so it will give us a count for every column, every row in the G ranges. So we're going to pass it our non-overlapping exons. We'll give it, here I think I'm just giving it one BAM file, but I could give it the BAM file list, my BAMs. I think this is just called my BAM here. I'm going to tell it to ignore strand equals true, and that means it's strandless. Your genomic center can tell you this. Finally, I need to set a new feature just for this counting, and that is interfeature equals false. Let me see if I've got any slides on that. We can see why. So interfeature equals false is going to allow me to count reads which are in multiple exons. If a read belongs to multiple genes, so this read could be in, goes from one gene to another, I can't really be sure where that belongs. Okay? I wouldn't expect a read to go from one gene to another and still believe it. But in this case, I could very much have a read which covers a little bit of this exon and a little bit of this exon. Right? I could very much have a read which covers a little bit of this exon and a little bit of this exon. Right? So I don't want to throw them away. And we're going to assign them to both exons. It's just what we have. So setting interfeature equals false means it's not going to throw away reads which go across features. Okay. That is always set to true by default, and for RNA-seq, you definitely want that. If you set this to true or you forget to set it, you're going to have barely any reads counted, because most reads are long enough now that they cover multiple exons. Reads can be 100, 200 base pairs long. So once we run that, we're going to end up with, hopefully, an object we recognize, a range summarized experiment object. A range summarized experiment object is basically a complex matrix. It's got the matrix of counts in the assay, but it has additionally row data, which will be our here, the G ranges we counted over. That's the ranged part of the summarized experiment. And it also has this col data. Col data would be things like the condition. So actually, when you're doing summarize overlaps for genes, right? So if I was to count over genes, I'm going to provide a G ranges list here. So then what it's going to do is it doesn't worry about interfeature within the G ranges list element. It just tries to count all the reads in that G ranges list over those exons. But what you don't want then is a read which goes from one gene to another. And it's actually what often happens, if I can just open up IGV. Does that answer your question? Yeah. Cool. I'm just going to open up IGV to give you the problem example there. Yeah, if I can find IGV, hang on. Hey Siri, can you open IGV? It's nice when that works, rather than sends me to like a TV channel or something random. Load from file, new session. Let's go. Actually, Chicken's got a lot of these examples. I need to load a GTF. So human, H38. So here we have like a really tight collection of genes, and this is the Hox cluster. And like developmental genes tend to be all right next to each other. They like to be regulated in like what we call tags or something like this, right? Regulatory domains. The issue is that quite often you will get some reads going from, say, HOX2 here, which start to spill over this tiny gap into whatever this one is, HOX ARM. Okay. And that's just because, you know, transcription doesn't die straight at the end of a gene. It goes on a little bit more. We tend to pick it up. What happens if you do this interfeature equals false is you start to capture the tail of this in here, and then you start to count it. So like reads which would belong to this gene, and just because this gene's piled on a little bit further, expression didn't truly stop here, it starts to get picked up in a neighboring gene. But if that read is spanning two, and you say interfeature equals true, it's going to throw away any reads which touch this gene and that gene. That's kind of one of the ways we can avoid things like this. Another time you'll do interfeature equals false is when you do things like, and you're going to see this in single cell, but you see it a lot from places like the Heinz lab here, Atom Lab. Anyone working on neurons, what you tend to do is extract the nucleus, and you will do nuclear RNA-seq, effectively use an antibody for the nucleus to get a particular cell type. That is unspliced, right? Which is a big problem because often genes overlap each other. I'm not sure if we've got an example here in the Hox region again, but zoom out a bit. Sometimes you'll get genes within other genes. So maybe you do have some examples here. So, you know, HOXA10 is within this other gene here, right? The only way, if I have pre-spliced mRNA, which is from nuclear because it hasn't been spliced yet, I will get signal across the whole length of the gene. And then I can't tell, is it from this gene or that gene or this gene, or then I just assign it to all of them. I will worry about that later on in my analysis. Okay, look, these genes all overlap. I have to go review it myself. So nuclear RNA-seq, and then we have the same problem now with single cell. They often sort single nuclear RNA-seq. We have to think about it the same. Cool. So our range summarized experiment then is just like all the other ones we've seen. We can still extract the matrix we want. So the actual counts by using assay. And here we got, not sensible necessarily row names, but this is the gene, this is the exonic part. But we have counts across all our samples. And if we look in the row ranges, this is exactly what we counted over. We have our disjoint exons, and actually finally we can see a disjoint exon here with this gene. This exon goes to this part, and then one base pair later there's another exon. Right? So it's probably split them up for us. So we have the range summarized experiment object here, but a lot more rows, 269,000 rows. So this is going to take a long time to process. And I advise, this has parallelization built in. I advise you do this on HPC. So previously we've been looking for things like this. Lots of signal here, no signal here, changes in overall gene expression. Every exon is gone or changed. Now we want to look for things like this. And this is just 2 cell lines, but you can see this gene, exons look pretty similarly expressed, but then this like extended transcript here, exons are only expressed in this transcript variant, and HeLa cells not yet. And if we were doing it at an exon level, we would just detect that these exons are different between the conditions, but the remaining genes aren't. So, this is why we then do disjoint, so that we can have times where we can actually, hard to say here, but this is, I guess, just that much more of a complex example. But then we're going to try and count over these disjoint exons. And then, as I said earlier on, this may allow us to try and pull apart which transcripts are actually changing. This example is pretty straightforward, though. Anything which changes in these exons, we're going to believe then it's associated with that transcript. So one of the most used tools for this is DEXseq. So this is written by the same people who did the original DSeq, not DSeq 2, but then DEXseq is well implemented with DSeq 2 now. So it uses the same algorithm under the hood as DSeq 2, but it's going to run an additional test to effectively say, does this exon change differently between conditions compared to all the exons? So, all the exons on average may not change, but this exon changes more. DEXseq is a little slow when it's fitting its models, but it has options to do parallelization. They are still slow. Voom and Lima are much, much faster, but it provides a less informative output. I'm going to show you can do some stuff with DEXseq output, which just makes it really easy to interpret the result. These are faster. I just personally can't really find a useful way to work with them. So DEXseq, like all bioconductors, it kind of follows the similar set of setup. We're going to be able to work with our range summarized experiment object. But first, just like we did when we were working with DSeq, we just need to add a little bit more information to this. And the information we need to add is our conditions. So what is our actual groups here? So to do that, we'll just create a data frame, and I'll assign a condition. And this is we had 2 siRNAs in the experiment. This is just one of the siRNAs, PTB1. And then our other experiment is senescence. So this is senescence plus this siRNA, and this is just senescence. Give it some row names so that we can match with the sample names in the range summarized experiment object already, the column names. And then with that, I can then attach that back to our matrix later on, the range summarized experiment. So previously we've used DSeq dataset from matrix, and we can use the DEXseq data set in a similar manner. So as with this DEXseq data set from matrix, it requires a count matrix, a G ranges, gene IDs, and slightly differently here, it also needs exon IDs. So it's going to need to be able to tie gene IDs to exon IDs to work out these exons change more than the gene. We're also going to need to specify a slightly strange design. So previously we've set by the group of interest. In this case, we're actually going to look for a sample effect, an exon effect, and then the interaction of our group of interest with exon. We're never going to need to change this, right? So we're not going to get more complex. This is just allowing us to see changes in gene expression associated to a sample, changes associated to an exon. What we're really interested in is when an exon changes in relation to the condition. So this is always the setup. I just wanted to show you what it looks like. So then, in order to actually run our DEXseq experiment, then we can just use the DEXseq data set constructor. Once we've loaded the library, give it our count matrix, give it our metadata, so the col data, tell it the design, tell it now the feature ID, so that's our exon IDs here, and then we can tell it the meta feature ID as group IDs, and we'll give it the gene IDs. Okay, so these two vectors here, which we're adding, just allow it to tie this exon as a member of this gene, and if that gene changes and the exon changes the same way, that's not interesting. If the exon changes and the gene doesn't, that is interesting. As we have already generated a range summarized experiment, we can actually use the DEXseq dataset from SE function, so summarized experiment. We just have the range summarized experiment version. To do that, all we need to do is provide to the DEXseq dataset from SE our senescence exon count, so the range summarized experiment we already had. So this object here, do I actually show that object? Yep, this object here. And then we need to specify, always we need to specify our design. Again, it's just always sample, exon, and then whatever our group information is here, we know that's condition, and then exon. Okay. I always found that a little weird, that DEXseq has DEXseq from dataset, DEXseq dataset, and accepts a summarized experiment object. This one goes a bit backwards, and DEXseq dataset needs the count matrix and all the parts, whereas this one from SE works a bit more like DEXseq does. Hi, Dave. So DEXseq, like I said, is very computationally expensive. I say fairly here, it's very computationally expensive. It takes a long time to run. So we can speed this up somewhat by removing exons that have, say, arbitrarily here, but this is generally followed, less than 10 reads and at least 2 samples. Okay, so our smallest sample group is 3. You could say it needs at least 10 reads and 3 samples, and that would mean in one sample group it would need to have at least 10 reads. But we're just going to apply that to the matrix. And for every here row, using apply, going by the rows, I'm just looking for when the total number of reads is greater than 10. It needs to happen in at least 2 samples. We can use that then to filter our senescence exon counts, and this is a significant filter. We've gone from 200,000 exons down to only 100,000. But all those exons we've thrown away have barely any counts over them, and they wouldn't have been interesting for our test anyway. Having done that filter, I'm then going to run the DEXseq dataset from SE, give it everything it needs, our range summarized experiment of exon counts, our design, sample exon condition based on what was in our senescence exon and counts, and then exon. everything, just like with DSeq, it has its own little pipeline, and we can run the DEXseq pipeline. If we want to do it in separate steps, we could do estimate size factors. This is our normalization factor. Estimate dispersions. This is fitting our mean variance relationship. Lower the mean, the more variable. Higher the mean, less variable. We can run this step here, which is our test for differential exon usage. And if you can remember when we did our DSeq LRT test for multiple groups last week, we compare our DSeq object to a reduced model. And the only thing we've changed in the model here is removed this interaction between condition and exon. Again, it's always going to be like this. You don't need to change the reduced model formula. And then we can estimate actual exon fold changes. So how much does each exon change between conditions by running this final line? Just tell it which condition to fit to. We only have one. So we'll do condition. Once we have processed our results through this, we can run just like we had DSeq results, we have DEXseq results. We're only ever going to have one result type here though, right? Because we're always comparing our full model to this reduced model. And then like most results, I will order it by p-value. So I have the most significant at the top and the least significant at the bottom. Just like with DSeq2, we don't need to run every single step separately. You can just run DEXseq, give it the DDX object, so our pre-normalized object, and it will run everything here in one go and just output a result. We can then look at ourselves. Let me just have a little look at DEXseq function here. I think I've got it installed. Install it. I just wanted to show you quickly one thing which we're not showing here is DEXseq allows for parallelization. It's one of those options. So spelt it wrong. It's nice that when you do this, Bioconductor gives you that nice message. Probably spelled this wrong. Did you mean that? Yes, I did. Okay. So then DEXseq, so look at the function. You should see one of its options here is BP param. I'm not sure if we didn't really go into that much detail, but BioC parallel had this function multi-core param. And then we had seen previously, if we just do require BioC parallel, you can always multi-core param. How many workers we have available. I spell multi-core workers right there. Should go off and tell us how many workers we had, but actually I think my computer's don't throw it. So I got 14 cores, and then I could, if I just set register multi-core param, I could actually set the number of workers to how many cores I want to use. So that's just telling my computer to get ready for parallelization. And now if I run this DEXseq command, it will go off and start parallelizing when it does the normalizations and all its various tests. Boy, my computer's really slowing down, so something's happening in the background. If you actually look at the results, it contains a little bit more information than we had from DSeq. So this is our DEXseq result object, straight back from this DEXseq function. And you can see it has the group ID, so that's our gene itself. It has the feature ID, so our actual exon ID, and then the information on the base means, the dispersion, the stat, the p-value. And actually then it has some really nice information. It has the mean expression in senescence versus SH-PTB1 knockout. It has the log fold change. And then it contains within this table a column which contains the G ranges, so the actual regions I counted over. The actual original count matrix is held in one column here, so I can extract that too. And then really usefully, if you want to start to tie this back to transcripts, you also have the transcripts which this exon is a member of. Okay, so it belongs to this gene, but it's also a member of these transcripts. It can also be converted into a data frame, which is very useful if you just want to write it out, but this will act a little differently to a DSeq object. If I do as.dataframe on it, you can see it expands. This column is where we had the genomics data as a G ranges, and we had a matrix here. When I turn this into a standard data frame by doing as.dataframe, I now have multiple columns for the genomic ranges, and I have multiple columns for my count data. That's still a very useful table. A really nice feature about DEXseq is that the guy did think somewhat about how you're going to then plot it and review this. We have this really nice function, plot DEXseq. We just need to tell it the gene of interest, which we want to plot our differential exon usage over. And you can tell, well, this was the top one, 57142. So if I say plot DEXseq, DXR1, so that's our DEXseq object, and I give it the name of the entree's gene ID, I then get kind of a sensible-ish table. The ordering along the Y isn't necessarily sensible, but here's the gene model, and it's telling me this exon and maybe the exon next to it were differentially regulated compared to the rest of the gene. I haven't got the color scale here, so I can't tell you which one, but one of the samples had a much higher expression for this exon, slightly less for this exon, but all the other exons are basically identical. So there's some differential exon usage here, and that would maintain to this particular exon. From here, I could also look up the G ranges and go review this in IGV. Because it's telling me it starts there and ends there, this particular exon, which is differentially regulated. I think if I went and looked at it, this is the example for the first one. It's really clear. I mean, I think this exon's a little less clear. It's just about there, but that's very clear. This is only used in the PTB1 silent sample versus this one. So that's a nice example. We had a slightly more complex example, I think, for the second one. But I know we didn't. Sorry. If you want to get a bit more details on actually the transcripts for this, we can also just put display transcripts equals true on this plot DEXseq function. You can see then it shows us our kind of meta transcript, all the transcripts squished on top of each other there. But then it shows us the individual models. So we could say, okay, well, this differential comes from probably these two transcripts. And it's these transcripts which are not changing. Like I said, looks good in DEXseq here. It does get hellishly complex, though, with some of these. Here's an example of one which is really hard to interpret. And if you do plot DEXseq, I think this is the second hit. But you can see here, red kind of flips over. It's different here, and then red's very much lower here. And then they become equal for some of them. Okay, so it's much more complex. If you go into IGV, we can start to see what's really happening. And it's these exons here, which I guess would have been, I think this is backwards. It's going the other way. Would have been these ones here. Okay, but we can also go back into the actual table and see which exons it is, and then go and review those in IGV. But certainly the top two hits pulled out some really nice examples where you have this exon, I think, is also differentially used. These ones are gained, and if we look here carefully, there's an exon which is lost. Right. Going back to this, though, it's going to be complex to work out which transcript is actually probably accounting for that change. But at least we know that exon in particular was lost here, and these exons were gained. If we want to, we can also make an HTML report from this, and this is actually quite useful. I wonder if I've got it, if this link works. Nope. I think it's downloadable in our file. So if we do DEXseq HTML, and we just tell it the gene, it actually produces like a nice little report for us. So let me see if I can quickly dig that out. My computer might have collapsed on itself. Sorry, my computer's going too slow. I'll try and dig that out at the end. But you can also run the example method. If you go to their example, you can also run that and get this nice little HTML. The nice thing about the HTML is it actually runs all these different plottings for you and gives you tabs you can kind of step through and review the results. One thing we might want to do is actually get a value for how well, so at the moment, these p-values are all based on an exon. Right, so this exon is differentially used, and we have a p-value associated to it and a stat associated to it. We might actually want to summarize this to the gene level. And DEXseq has a way of basically producing an adjusted p-value of the gene level for all your genes. All we need to do is pass it the DEXseq object we produced. We give it to the per gene Q value, and then it will take into account that every gene has different number of exons. Right? And if you have more exons in a gene, you're more likely to be differential exon usage. It's just a matter of probability. So from this, it returns a vector in the same length, in the same order as your DEXseq DEXR object. So in the same order as this, it's going to return to you your gene-related Q value. If we just look at the first 10 here, they're all 1, so nothing had passed. But we can just make a data frame from that, give it the names of our genes as the row names. So here it's going to turn this into a data frame of 1 column, p-value being the column, the row names being these names here. We will merge that with our DX data frame, our DSeq results. We can order it by here, the gene Q value first, and then the p value. So it's going to order it first by whether a gene changed and whether an exon changed in that. And now we can see we have our top 2 hits, which I think are the top 2 we were looking at. Maybe. That's everything we're going to cover in terms of RNA-seq. I think we've done pretty well for time. We have some exercises here, and they're super simple. And the reason they're super simple, I think you're basically just going to load in the results, is because DEXseq takes a long time. Even within an hour, you're going to struggle to get this finished. So I'm just giving you some results to look at and review here. You can do some of these plots. In this case, it's kind of an interesting one, because actually the gene did change, but this exon changed less than everything else. Right, and then you have another one here, just to get you some practice on this. I'm going to remind you that later in the course, we have, this is a nice way of visualizing differential exon usage straight away. Just a reminder, later in the course, I can't really jump around so much at the moment. I'm going to show you how you can visualize much better in like an IGV style plot inside R. So these are cool, but we're going to make some real nice ones later on as well. Okay, so I'm going to leave you for an hour or so now, just to run through these. If you want to go home and pick up the rest of the course from there, please feel free. I'm going to stop share. I'll pause my recording. Share my screen. Yes, class. Okay. So we have got through a lot of stuff now. We've done most of the basics, and we had to go at RNA-seq. The next technology we're going to look at for high throughput sequencing and how we can analyze it is ATAC-seq. So anyone here going to be doing ATAC-seq? You guys are going to be doing everything just as well. So ATAC-seq is a great method, and it's kind of developed more recently than most of the high throughput sequencing methods we use. And actually, it's one of the ones I had the joy of discovering how to analyze when I came to Rockefeller. So I had to kind of pick this up and learn this when I came. And what we did is I actually hosted a little class on there we go. Actually, the top hit. So we did a little class on New York R and Bioconductor. I'm trying to find the actual link to it now. There we go. Gosh, it's so complex just to find it. Anyway, so I took a lot of the public data. So I took the original course, the original ATAC-seq data, picked a little bit on Christina Leslie again, and we took some of her data again, just so that we can get a good idea what the original ATAC-seq looked like, what the modern day ATAC-seq looks like, and really dig down into low level looking at these metrics. So that's what we're going to cover today. I've split it up into 4 or 3 sections. A lot of this we've already covered, you know, basics, but now we're applying it, so hopefully we can put it into some context. So ATAC-seq, I think it stands for Assay for Transposase Accessible Chromatin Using Sequencing, uses a transposase to efficiently fragment accessible DNA prior to sequencing. So this should provide a map of accessible open chromatin genome-wide. So I'm not sure if anyone, I'm not sure how much of your epigenetics you know yet, but obviously accessible chromatin is chromatin which is not compacted. And actually ATAC-seq is really going for very open chromatin. And from this method, by finding regions of open chromatin, maybe differential regions of open chromatin, we can then make assumptions about what sits in that open chromatin. And that can be in combinations with ChIP-seq or by using motif hunting. And actually, there are a lot of tools about finding open regions of chromatin. If it's differential and it's only in one condition, maybe it's interesting. And then we can look at what the motifs are underneath. In contrast to other techniques which promise to do something similar, ATAC-seq had a lot of advantages. And one of the major advantages, I'm not in the lab, so it doesn't affect me, is that you could do it in a day, which is very, very appealing, especially if you're doing large numbers. Maybe you can do 2 or 3 batches in a day. And you need a very low input material. So compared to things like RNAseq, which is now probably a dead technology, but DNA-seq too, you need a lot more material for that. So, you know, I think a lot of you would know this, but given closed chromatin and open chromatin, euchromatin, we can use this TN5 transposase, and what it does is it breaks up the DNA in the areas of open chromatin where it can go. And this really means we're going to get lots of different types of fragments from our sample. But really, we're going to be interested in 2 types of fragments. Fragments of totally open DNA, so regions like this, and regions around nucleosomes. So if you look here, this nucleosome isn't compacted into closed chromatin. It is open, it's an open chromatin, and we will get with this TF5 transposase cuts either side of this nucleosome, this histone component the DNA is wrapped around, and then eventually we're going to end up sequencing that. Right? So there were, as I said, this is a great image. I've taken it from someone else, so I need to probably create my own version of this image. But just to give us an idea of the different types of techniques doing something similar, RNAseq was the enzymatic digestion of open chromatin, really around transcription factor binding sites. And if we have our little model of DNA here, we have our nucleosomes, our histone complexes, and then we have probably, let's say, an enhancer around a transcriptional start site. We have some open chromatin, and we have a transcription factor sitting in the middle of this. When we did RNAseq, we would actually extract sequence from around this open region and around this transcription factor. And we could use the size of the fragment, right? This fragment's bigger than these smaller fragments to give us an idea of where the transcription factor is. RNAseq did something slightly opposite. It was really looking for breaking up the nucleosomes. You didn't get signal from this open region in the middle, but you got signal around nucleosomes across your whole genome. That's expensive, effectively mapping your whole genome with RNAseq. So something my wife tried to do, and it just costs a lot of money. TAC-seq actually allows you a method to simultaneously extract signal from both this open region between nucleosomes, and then to a lesser extent, you're going to get reads from around nucleosomes. And the more compact the chromatin is, the less reads you get. So we should be able to, with this, get the position of nucleosomes, maybe, right, from this type of signal, but also get a map of where the open chromatin is, where our transcription factors may be binding. We might even get some signal kind of reflecting the area which was protected because a transcription factor was bound to the DNA. So this is all cutting around areas which aren't covered or protected by other features on the DNA. So we're going to cover the basics today, TAC-seq basics, and we're going to look a little bit post-alignment too at some quality control we can get from some simple tools. I actually think we're going to plot this at the beginning of our next session. So it's not. So like I said, when I first put this course together, I spent a lot of time making sure we had some great data. So I think I took it from 3 data sets. This is the original TAC-seq paper, I believe, by Buenstro and Greenleaf. He's gone off and made his own lab. Buenstro does new stuff, and Greenleaf has maintained their tools in the TAC-seq. We can download this, and we could go probably here, which will be the ENA browser, and we can go download this one sample, or the samples associated to it. I've already done this, so we don't need to. Oh, that's the wrong place. We don't need to. The second data set is going to take from ENCODE. They have lots of data, and most importantly, they have 2 replicates for every group, so we like to use them too. And the third data set, again, is from Christina Leslie's lab. And if you remember, in the RNAseq, we were doing some of the same kind of experiment. So we were looking at T regulatory cells, naive, before they've been exposed to some stimulant, and then activated. They've been presented with the antigen. We can find FASTQ for those files here and here. We're actually going to be working with the aligned data. So I've already done what I'm going to present in the course, some of this data. But it's actually on ENCODE as well, I believe. It's just a much newer data set compared to some of the other data sets with it. So Bing Ren, maybe that's a while ago. Yeah, maybe. Same thing. So we need 3 pieces. We need some reference data. We're going to need a reference genome in FASTA format. We know where to get them from. We can use the BS Genome Bioconductor packages or your favorite FASTA file of genome sequence. Gene models, we can get those from the TXDB packages. We've seen that before. We can also get them from a GTF and turn it into a TXDB. And finally, we have something which we'll talk more about in our ChIP-seq. This is the blacklists. I'm not sure if anyone knows about the blacklists. It's fascinating. See if we can, what it pulls out if I go to the ENCODE study. See, maybe I didn't follow quite the right link. Try it again. Nope, didn't go. Do you have any history on it or anything, or a PubMed link maybe? Okay. So I did a lot of work with these actually in my first years as a bioinformatician because I was very big on ChIP-seq, my speciality. We wrote a little paper on this too. And really it was a fantastic piece of work by a number of labs, but Anshul Kandarji, I think we can probably say, was the guy who really put it all together. And what they did is fascinating, and we can actually see it better in the ChIP-seq. But what they did is trawl through all the ChIP-seq, which was coming out of the ENCODE study, and they started to see that there were regions of the genome which just had signal no matter what you were trying to chip for. So if you're looking at MYC, CTF, polymerase chip, there was always signal there. And actually, it turns out from our work and other people's work, it turns out this is basically to do with repeat expansion. It's because your genome's got bigger than you think it is. And if a genome which you think is 100 base pairs long is actually 100,000 base pairs long, when you go to get a random sampling of reads over that region, randomly sampling over 100, you're not going to get many reads. 100,000, by chance, you get a lot of reads. And then when we realigned it back to the genome we understand, we end up with this tiny bit of region, 100 base pairs, which is probably repeated 1,000 times in the genome. And we map to that one region and we just get a pile up of reads. So he went through and he actually paid a lot of attention and manually effectively selected these regions, and now they're filtered out from most ChIP-seq. We're going to cover it more in the ChIP-seq course, because the reality is, I used to work in Cancer Research UK in the UK, obvious. But if you work in cancer cell lines, those cell lines change all the time. The blacklist changes all the time. And you need tools to effectively discover your own blacklist. So there's something called gray list, and we'll cover that in future sessions. But using the ENCODE's derived blacklist, we're going to do a good job about removing much of this artifact. So the blacklist is really just per genome, a set of regions where we believe we have artifact signal, and we're just going to filter them. So to get started today, we need a BAM file and binary files, obviously. We're going to show you how you align them, but I've already produced them, and you can download them from here and here directly. If you want to get, and this has a little bit more because I did this originally as a separate course, but if you want to get all the files you could possibly need to follow along, you can click here. If you want to get all the intermediate files and everything, you can click here. And that's going to get all the BAM files and everything you would possibly produce in this class. Bigwigs, if you just want to follow along in IGV, you can download from here too. Now we can get started. So prior to alignment, we always recommend checking your FASTQ quality. Some basic QC checks may identify bias in your data, so you have GC bias. So we'd already seen this kind of in our bioconductor intros. We saw it in our RNAseq, and we're going to do it again in our ChIP-seq. So I thought I could save us some time, and we'll hold off and go look at the FASTQ quality checks in our ChIP-seq. In reality, you can run FastQC, just see those plots, review those plots we talked about, and you'll be okay if nothing looks too strange. So we're going to work with Greenleaf actually through this course because it's a really great example. We have his plots from his paper, so we can compare everything we do, and we can make sure that we can recreate the original ATAC-seq. First thing we need to do is create something we can align to. We've done this before, we can do it again now. We're going to create a FASTA file of the major chromosomes, and we're going to write to that, and that will be our input for our index. So we'll just do library, BS genome, H sapiens, UCSCMM10, HG19. So that will load this object into memory. Then going to quickly make a vector of all the chromosomes we're interested in. I loop through that vector, And for every time I go through the loop, I extract one of the chromosome sequences, and that comes back into a list. With that list, I will then provide names, because the names I provide, which will be chromosome 1, 2, 21, these are what goes in the FASTA header line. So I need to provide names. Once I've got my named list, and each element is a DNA string, I can then turn that into a DNA string set. And I can write that out to file here with writex string set, and that will provide a FASTA file, which is all the sequences for the human genome. If, for whatever reason, you don't want to use the FASTA files from inside the BS genome object, you can just start from whatever the FASTA file you have here. And that includes, downloading it from Ensemble or iGenome. We then use our build index function from the subread package. So we're building, this is just our FASTA file. We then need to build an index to align to. And to build an index, we're going to use the R subread function build index. We'll give it our FASTA file, which we just created. So from here, we're going to tell it the name of the index to create. And then that's going to create a file called whatever this, main cur. And then we'll set here now a few features to control memory. So first off, we'll tell it index split. And that means it's going to try and chunk up our index into separate chromosomes or separate regions. And then we'll set memory, and this is going to be the amount of megabytes used both in the indexing, but also the alignment. Right, so you set this low. When you come to alignment, you're also going to save memory. If you set this high, it's going to get faster, but you're going to need more memory. So I think by default, it sets this at 8,000, but you always need to set index split to be true or false. So here, if we set it to true, it's actually going to try and reduce our memory. If you set that to false, this memory parameter has no effect. So now we have an index. We can align our TAC-seq reads. But a TAC-seq data is a little different to what we've been looking at previously. Right? So RNA-seq data can be single or paired end. So can a TAC-seq data, but more often than not, it is paired end. And there's a real reason, as you'll see as we go through, why paired end sequencing became kind of popular for a TAC-seq. So for paired end sequencing, the most likely, I mean, I think 99.99% of the time, going to see your reads kind of separated like this. So you'll have a sample name. So this is the name from SRA. But then you'll have either an _1, _2, or an _R1, _R2, and that just tells you that these reads are paired. This is the first read in the pair. This is the second read in the pair. Okay. So paired, well, these two matched paired end read files should contain the same number of reads. They may not do. If you've got them straight from your genome center, they will do. Someone may have filtered or changed your FASTQ before they get to you. Okay, so that is a possibility. So you might want to check that. But we can get an idea of what's happening inside our FASTQ pretty quickly just by loading the short read library. So short read allows us to handle FASTQ data. I can then read the first pair here, the second pair here, right? And I know it's the first pair by _R1, _R2. And now if I actually look at the ID for my reads using the short read ID function, I just look at the first two read names. And it tells me a little bit about the width, whatever, but I can see here the machine, flow cell, the position. And then here I've got this 1. If I look here, I've got the equivalent 2. And then this name here should match everything else in this file. Oh, sorry, in the first ID. So this ID and this ID match. That means they were paired. Can you see that? So this name here and this name here, they do match, right? Yeah. Yeah, the only difference really is this, read 1, read 2. So these reads come from the same fragment, and they're actually reading from either end of the fragment. So we can look at that here. If we look at, this is the actual fragment we're reading, okay? There's a little bit of a confusion when you go to your genome center. You're going to have difference between fragment sizes. Remember, when you go to your genome center and you get that final size of your fragment, that often includes the Illumina adapters to the end of your fragments, and they are just required for sequencing. That's where we start sequencing from. But it's this insert, this fragment, which is our arbiter sequence. So this is these broken up bits of DNA here. Those are what we're sequencing. And then we'll add the adapters, and then read 1 will start from one side and read into the read. And then in the sequencing, we'll flip this, and then we'll read from the other side, going backwards across the fragment with read 2. So you can imagine quite often read 1 and read 2 may overlap in the middle, and they're short enough the fragment. We'll get an idea of this. We should have sequence which matches. But this allows us, this gives us a lot of power in sequencing, because I can align this one, and then I can align this one separately. But then by knowing that they're from the same pair, there's an expected distance between them, right? Something between 0 and probably 3,000 or something. The distance between these two reads will also allow me to map better. So then we have what we would call the fragment size is often considered with the adapters. That's often what you'll see when they run a bioanalyzer or an electrophoresis gel. You'll get the size, and it will include the adapter. And then the actual thing we're sequencing is often the insert, and we'll refer to the size of that as the insert size. Cool. Just the read 1. So it's this. So that bit in black here, that's what we're reporting for our first read. And then the second read will report this going this way, right? But backwards. Oh, because these are the read names, right? So this read name is the same across the two, but the sequence will be different. It may overlap in the middle, and then you'll be able to tell this. But yeah, this bit here is the ID. If we did seq read 1, we would get the sequence for it. And actually, we wrote some software which, when they overlap, it allows you to make one long super read, and that's useful for it. We can use that standard alignment of DNA as we did for ChIP-seq, but we need to increase the maximum allowed fragment length. So what I was saying here about the maximum distance between read 1 and read 2 can often be a little bit bigger than we might expect for a TAC-seq. So that's one parameter we want to change in our normal alignment versus what we saw in the standard bioconductor. So I'll do align, and I'm going to set the BS genome index we want to align against. I'll tell it, I've got 2 reads now, so I'll tell it read 1 file is read 1, read 2 file is read 2. And that's just the names of the files here. Output file, we're going to call it attack 50. This is the name of the sample on GEO. I'm going to tell it type equals 1. Type 1 means DNA here. We didn't have to change that because we used subjunct for alignment previously. We're going to tell it number of threads. So strangely, subread isn't quite bioconductor in its parallelization. So we can't use BioC parallel to set it up, but we can say number of thread equals 2. We'll say unique equals true, so it's only going to report uniquely mapping reads. And then we're going to say max length equals 2000. Okay. There is a min fragment length as well, but we don't need it here. But we're saying consider a read properly paired if it lands within that distance of its pair. Read 1 and read 2. We can also use our bow tie to do the same thing. So just to show you that, and a lot of people like bow tie 2. To do that was very similar. We just do library rbotie2. We can provide the FASTA file we produced to references. And then we just tell it the name of the index we want to make. So here, the same as previously with underscore bowtie 2. And then if I want to do an alignment, the pain with bowtie 2 in R is that I need to unzip the FASTQ files. It doesn't take the zipped files, so I'll use gumzip function, which will just unzip a file to just the FASTQ. And then if I wanted to use this aligner, I will then use the bowtie 2 function, provide the index, provide the SAM output. So it's going to make a SAM file rather than a BAM file. And then I will provide the links to the 2 FASTQ I just gzipped in this step. We don't need to specify a maximum fragment length in bowtie 2. It's not going to cut off reads if they're too far away. And then we can just do, on our SAM file, we can just do as BAM, give it the path to our SAM file, and it will just update that file and produce a dot. And so either way, we can either use standard R subread, which I prefer, I understand a bit better, or we could use bowtie 2, which is also a very popular tool for RNAseq. I think they use bowtie 2 in the paper, which is why I use bowtie 2 here. Not sure if we've used this before. Have we used unlink in the class before? Probably deliberately not. Unlink means delete, so be careful with it. So what we'll usually do is, this is your script. I'm just putting it here to make sure that when you've done your alignment and you've made your BAM file, remember to delete the SAM files and delete these FASTQ files, because they are going to be huge. SAM file is wasted space. You have the same information once you've done as BAM. It's created a BAM file. So just clean up your scripts for bowtie 2. Following alignment, we want to probably do our ordering and sorting. So here we're going to use our SAM tools again. We'll just create the path we want to our sorted BAM file. And then I'm going to do sort BAM. And I'm going to use my out file from probably here. And I'm going to pass that now to sorted BAM. And then we'll give it what we want to create. I'm just doing this G sub because I'm removing the .bam extension. If you remember, when you run sorted BAM, sort BAM, it doesn't want to have the path you to the file you're outputting should be the prefix. It should never end in .bam. Otherwise, it's going to create a file called sorted bam.bam. We can then index our file, and indexing is super important because it makes our file read access, random read access, so you can read from anywhere in your file. It means it will work in IGV, and it also means we can get those very quick stats from IDX stats. We don't need to read the BAM file, we can just read the index to know how many reads we have. So there are some really simple QC we can start to do, and I think this first one is the one in the paper and is the first thing you ever need to check in RNAseq, and it will tell you whether your experiment has failed by a very common problem with RNAseq. So we need to check the distribution of mapped reads across your chromosomes. We know IDX stats doesn't provide much information, but it does tell us how many reads map to each chromosome. So we can use that now because we ran index BAM here on our sorted file. So we can just load the RSIM tools library. It should have been loaded here anyway. And we'll do IDX stats BAM on our sorted BAM file. And that will produce for us a data frame of all the chromosomes and the number of reads mapping to them. Okay, so show you what that looks like. Guess I have a BAM file somewhere here. Session, set working directory. Session, set working directory, choose directory. Oh, actually, maybe I didn't download the Sure, I've got one in here somewhere. Library of samples. And then, cardiac stats, BAM, data. Let's do heart. Heart. Oh. I don't know what I just did, but I just blew up my arm. So maybe we'll. I get the feeling that that wasn't a complete BAM file. So maybe I'll try just the demo for now. If you ever get that, like your arse suddenly breaks like that, you've either hit your memory issue or it's because something you're using doesn't necessarily error well. And it's usually C++. So it's passed it down to C++ and some memory's going around. No big consequence, just to restart the file again, I guess. Sorry, I was looking for an example of IDX stats BAM, but I don't think we have one in there. Things here, so. Index BAM. Oh. That was painful, but so we have seq names, so it tells us the names of the chromosomes, tells us the length, and importantly, it tells us how many reads were mapped on every chromosome. So that's really what we're after. And once we have that information as a data frame here, we can then pass that to ggplot and just get a very quick overview bar plot of where all our reads have gone. So if I do library ggplot, I'll do ggplot map reads, I'll do rows here will be the seq names, the chromosomes, the number of mapped reads, I'll fill them in by the chromosome name. We'll do G on bar, but I'm not asking it to calculate the size of the bar, so it's stat equals identity, and then I'll just do toward flip, which just rotates the flip plot. So plus toward flip just literally rotates your plot 90 degrees. And already we can see something really weird. Right? If we look here, chromosomes are in order of how big they are. So it makes sense that chromosome 1 was the biggest up until around here, chromosome X. Chromosome X is actually quite big. But most of the reads have gone to the mitochondrial chromosome. Right? This was commented on in that original paper. We have a lot of mitochondrial reads, but it was most of the reads. Their lab has managed to clean this up. And there's things like RNAseq, which reduces this greatly, this amount of mitochondrial reads. I can tell you it's still high though. So you still are spending a lot of money on this mitochondrial read. And actually for the rest of ATAC-seq analysis, 99.9999% of analysis, we throw away the chromosome. It's a circular chromosome. It's very hard to analyze for mitochondria. Right? But that's a lot of reads wasted. So this is the first thing you can check to see if your ATAC-seq worked. It's the first thing people ask me to check for them. And then hopefully with the new methods, OmniATAC, etc., you shouldn't see this much of a problem. It's also a QC metric for single cell attack as well. So now we have processed the Greenleaf ATAC-seq PED-end data. Now we can start to work on the alignments. One thing we want to look at pretty quickly is the expected fragment length. Right? So given that we know we're going to be breaking up around nucleosomes and we're going to be doing an open chromatin, we can expect to see fragment lengths the size of open chromatin in there. And we can expect to see fragments the size of nucleosomes in there. So just to remind us, that's because we should have regions coming from around nucleosomes. We should also have regions coming from inside. Well, this is the nucleosome sized one here, from inside open regions. So these are also a very important QC for us, and they were important QC for that original paper. One thing we're going to do right from the start is going to set our flags to only read properly paired data. So we'll do library genomic alignments, because that's where all our tools to handle BAM files are. And we'll set a flag from our scan BAM param. So we'll get scan BAM flag. And I'm going to set one flag, and I'm going to say a read needs to be properly paired. And that means it needs to fit within this range we set earlier on. 2000 was what we set as the maximum fragment. So now I will build, now I have scanban flag, I can then build my scanban param object, and I'll provide the flags I just created. I will tell it, I want to just look at one chromosome here, so I'm just going to look at chromosome 20 from the beginning until this is the length of chromosome 20. So I'm going to provide that for the which. And then what do I actually want to pull out? I'm going to get the Q name, I'm going to get the I size, and I'm going to get the map Q. Okay, so Q name is going to be the read name. Map Q will be the quality of alignment. And I size will be the insert size of our fragment. So how long is our actual fragment? Okay, so that's going to be, come back, this size. How long is this fragment we actually sequenced? Right? And we can find that out from knowing where read 1 and read 2 are lines in the genome. Okay, so now I can use my scanban param, and I've created a scanban param object. And then I will pass that to my readj alignments function, except now, previously we did readj alignments. This time I'm doing readj alignment pairs, giving it my sorted BAM file, and I'm telling it the parameters. I want this information. It's only over chromosome 22, and just give me properly paired reads. We get a GA alignment pairs class. We had GA alignments class before. And if we start to look at this, this is like an extension of our GA alignments we saw in our bioconductor classes, but also it's really like an extension of G ranges. Okay. And it's kind of cool. What it has is the chromosome it aligned to, the strand it was on, and then it has 2 ranges per row. And this is your read 1, where that mapped, and this is where your read 2 mapped. Okay, so it holds that all in one line for us. Okay. If we want to get into this and actually find out, if I want to look more carefully at read 1, I can extract my first read from my object of reads here. I can do first, and I will get the first read. Second, I will get the second read in the pair. So I'm basically extracting this read in the pair or this read in the pair. And then I can just look at read 2, and we can see we now have all this information I wanted. This looks much more like previous GA alignment object. I have a cigar, I have the Q width, the actual width, the start and the end, the junctions, but I also have the Q name, I have the map Q, so the quality score per read, and I have this size of the fragment, the I size per read. So one of the first things we're going to do is get some type of distribution of our scores. So do our reads look high quality mapping? We can extract that from our GA alignments object for either one of the reads. In this case, I'm going to do first and second separately. But we'll use mcols to get the metadata columns. So that's everything after these dashes, the 3 metadata columns. So I'm going to get the map Q. So I'll do mcols. And then I'll do $mapq to select the mapq column. I do that for each read separately, and I can just look, and we can see we have 40, 40. This is the top score we can get for subread. And then I can use our table function just to summarize how often do we see values in our list. So I'll give it the result from here, and then I can look at that here quickly, and I can see that 40 being a good score, most of our reads are 40, and then that drops off. Same for read 2. Read 1 and read 2 could have different qualities of alignment, right? And if we just do a quick plot using ggplot now here, we can see that generally this doesn't look so bad. We have lots of high quality reads, high quality alignments here. What I would be concerned about is if I saw lots of 0s or 6s in this case. That will tell me there's something wrong with my data. I see mostly good quality alignment. This is our max, so I'm pretty happy. Now we have these reads already in paired end data format in R. We can then retrieve this insert sizes, and this is something which is super useful. Again, if this is probably the second thing people ask me to check in a taxi. Since the properly paired reads will have the same insert size, so the distance from read 1 to read 2 is going to be the same as the distance from read 2 to read 1. We only need to look at one of the reads. Right? We know they're all properly paired, so I will just extract the reads from my first read from my GA alignments pairs object. That will give me this GA alignments object. And then for some reason, I've used element metadata here. It's the same as mcols. It's just another, this is probably deprecated. mcols is probably the new one. But effectively mcols, and I will extract now the insert size column. So just to show you that, I'm basically trying to get this column here. This can be positive or negative depending on the orientation of the read, how far away. You know, that doesn't matter to us too. We just want the absolute fragment size, insert size. So we'll do abs, and that turns any of these values into positive. And if I just look at the head, I can see, you know, my fragment lengths here. So then I'll do the same thing again. I will use table on this to get the distribution of fragment sizes. And we're starting here, the lowest being 50. That's the minimum fragment size. We didn't set that, but that was the minimum fragment size in our subread. 51, 52, 53, 54. And it would go all the way up until 2000. Right? So we'll have a range of different fragment sizes. But because we know a tax-seq should represent mononucleosome in part, we would hope that we will be able to see a peak around the length of mononucleosome covering fragments. So sorry to keep hopping back and forward. But this distance around a nucleosome, I believe, is like 137, 147 maybe. So we know how big, how much DNA can wrap around a nucleosome. Right? So by cutting either side of it, fragments which are basically that length, we'll assume came from nucleosomes. If they're packed too tightly together, we won't be able to get in between nucleosomes, but maybe we'll get around 2 nucleosomes. And then the distance of DNA, which is wrapped around them, will be twice. So we can also be able to find fragments which represent that. And that was actually the next thing which they did in their paper, and it's the next thing we can do to check quality. We just need to pass this now to ggplot. So we'll do library ggplot, data frame, and we'll give the insert, we'll create an insert size column of our fragment length sizes. I do names here as numeric because a name of a vector, even though these look like numerics, a name is always a string. You can't have integer or numeric names. So we have to convert that to as numeric, give the count, just to be sure I'm making that numeric too. And then I'll pass that to ggplot, where I will have the x being the insert size. So longer insert sizes going across the x, the frequency of the different insert sizes on the y, and we'll do geom_line. It's not super clear. It's about as clear as it was in the paper. We can see we have this big full spike here. And this is your open short fragments, open chromatin. And as we come down here, we can start to see this bump here. And if you look carefully, there's a bump there. Maybe there's a third bump there. Yeah, you really have to squint for that third bump. But it is there. And those would represent 1 nucleosome length, roughly around 147, 2 nucleosome lengths, 3 nucleosome lengths. Right. If you put that onto the log scale, it starts to become way more apparent. So if we just, same plot, plus scale y continuous, and we'll do log 2 here. If you look at that now, you can see mono, di, tri, 4, 5. Right? So you can really see the evidence of this. And this is just looking at 1 chromosome. This looks, I think, identical to what the Greenleaf paper pulled out. So that's great. We can reassure this method we've reconstructed using Bioconductor is the same. But what they like to add onto it is really they wanted to define nucleosome-free regions. So they said anything under 100 base pairs couldn't have come from around a nucleosome. It does leave a large region where they're just not explaining. 180 to 247, they defined as being, for sure, mononucleosome length fragments. And then they did that also for dinucleosome. I think they actually probably labeled trinucleosome in their paper too. Okay. So we can make a plot and we can do something similar to what I think they did in their paper. Oh, there you go. So kind of messy, but 1, 2, 3, 4. We see 1, 2, 3. Remember, theirs looks a little cleaner because they're using every chromosome. We're just looking at one of the smallest chromosomes. If I'm not sure I advise this much anymore, ENCODE standards have updated since I first started writing this course. And ENCODE now will say, don't split up your data. Just keep it as one big open chromatin-like data set. But theoretically, you could now split it up by taking your paired GA alignment pairs object and just subsetting it so that only the ones with insert sizes less than 100, we can consider them nucleosome-free signal. Those between the ranges dictated from Greenleaf will say mononuc, and those between these regions will be dinuc. So I guess really anything which is, say this is the length of a nucleosome, anything which is 147 or above, we could assume is at least from one nucleosome, right? Because if I was to cut the open region here, I would have that bit of DNA wrapped around the nucleosome. If I unwind it, it's 147 long, right? So everything here could be anywhere between 0, I guess, and 147. Right? We would consider everything up to 147 to be nucleosome free. Right? We know it's not from a nucleosome. We might start to make the assumption once it's 147 and above, it comes from a nucleosome. So then the question is, why do they limit it all the way down to 100 rather than 100 to 147, just to be sure? Right? They don't want any of this overlap because they really wanted to be sure that we can consider it open chromatin. Was that the question? Is why isn't it 147 and below rather than just? So yeah, I mean, if it was much bigger than 147, we would start to at least know that we're contaminating our open chromatin, nucleosome free, sorry, with fragments which could have come from nucleosome. The issue is that there could easily be fragments in here which are over 147. You know, we're just making that assumption that they're probably belonging to. You know, it's not an exact science here. It's not exact, but it does a pretty good approximation. What a lot of people do, you know, there will be ones which have come from nucleosome free which end up in other ones just because the size of the fragment happened to be too long. But you should be able to clean up your nuke free by getting rid of longer fragments, right? They can't have come from nucleosome surrounding a nucleosome at that point. So you're saying, so whereabouts would that be here? So like here, from here to here. Yeah, if that was all open, hopefully though, we would have had a chop here, and then this region for sure we would have got some chops in, right? Transposase would have got in. So I guess that's the idea. But there still could be, for whatever reason, you managed to collect a fragment which didn't chop anywhere, and you managed to sequence that. The idea is that by getting the transposase concentration right, you know, you're going to break up your DNA in a reasonably efficient manner. If you really lowered that, you would get no breaking up, and then you would end up with really long fragments. Quite common is that people over transposase their data. When I first did this course in this class, there were a lot of people going, oh, shit, my data doesn't look like that. And it's because in reality, if you over too high a concentration of transposases, leave it too long, you get rid of all these other fragments. You end up really with just these short. You manage to break up all your DNA. Okay. So we can then separate out our reads or our fragments and then make a nucleosome-free mononuke dinuke. And then with that, we could use our RTRAC layer and just export the reads as a BAM file using export, give it the GA alignments pairs object, tell it the file name you want to write to, and just give it format equals BAM. Okay? And that will produce an indexed BAM file. I think one of the last things we want to do to actually output something is make a bigwig. Okay? So ATAC-seq is a little different from RNAseq in that we were worried about splice junctions and stuff for RNAseq. For ATAC-seq, we're not. It should be contiguously aligned to the DNA. Okay. So we can get our GA alignments pairs. We already have them here. If we look at one row, we can see we have 2 ranges. A little trick I can do with GA alignment pairs is if I apply them, or if I apply the granges function to that pair, you can see what it does is actually collapse it back down to a fragment. So if this is read 1, and this is the read 2, when I say granges, it actually goes from the beginning of read 1 to the end of read 2. You can see that here. Right? So that's actually kind of useful, assuming they must be from the same fragment. Now I can reconstruct the fragments as a granges, and with that, I can start to do some further downstream analysis. So the first thing I'm going to do is try and get an idea of my redundancy. To do that, I'm going to take my fragments from here, and I'm just going to use the duplicated function to tell me how many of these are duplicated, and I will use sum to actually sum up the number. So duplicated will give me true, false, true if it's duplicated, false if it's not. Sum, it will add up all the trues as ones, and it will give me the total duplicated fragments. I can get the length of the granges will give me the total fragments. And then to get a duplication rate, I can do duplicated divided by total. And so that will give me a duplication rate. There's a word which ENCODE uses called a non-redundant fraction. And that is effectively 1 minus this duplication rate. So this number here, 0.74, whatever, that is your NRF. That's an ENCODE metric. And that's a good score. There's quickly another QC we can check. We hope that not much of our data is too duplicated. Right? Given the size of the genome and the amount of open chromatin there should be, we wouldn't expect too much duplication. We can then quickly write this out to a bigwig. We've already got the fragments, right? So we created fragments here, so we can actually make coverage from the fragments rather than the original reads. And then we'll just do coverage on that granges. I'm going to weight it, so normalize it to the total number of fragments we had. I'm going to put in a million there just so the numbers aren't too small. And then I'll export my newly created RLE list to this bigwig file, and then we can visualize that. Okay, so just as simple as going from 2 reads to 1 long read, which represents the fragment, and then creating coverage weighted by the number of fragments we have. So I think the final thing we're going to cover today, just enough time, is 2 more metrics. So ATAC-seq QC is written by a friend of ours, Zhu, we like her a lot. It's not the most memory efficient tool in the world. And that's just because ATAC-seq is hard to deal with. You have to hold a lot of memory, a lot of data in memory to do this. So I'm going to show you a little bit about ATAC-seq QC, sure she's very responsive if you write to her directly. But I warn you, it will take up a lot of memory. And you may never get to finish it on your laptop. It's just doing a lot. Two really useful metrics from this are PBC1 and PBC2. So this is your PCR bottleneck coefficient 1 and 2. And these are, again, originate from AttackSeq, but have become, sorry, originate from ChIP-seq, but have become essential for AttackSeq. Before I jump into this, another thing you can do is I showed you how to sample BAM files. So actually, you can often sample a BAM file down to 10 million reads and I run this tool, AttackSeq QC. It will work a lot easier. If you've got 100 million reads and you want to run this on your laptop, it's going to struggle. So they have this kind of workhorse function, BAM QC. So you load the library, a library attack QC, and you can just provide the BAM file you want to process for attack QC to the function BAM QC, and it will give you an object containing some useful QC metrics. So like here, I've just filtered down to chromosome 17 to save us some time. Effectively, this gives us back a list, and we can see the names of the elements or slots within our list. Some of them we've already kind of looked at. Mitochondrial rate, we can calculate that ourselves very easily. Duplication rate, proper paired rate, right? And that relates to having how many fragments are within your insert size. Non-redundant fraction, we know is duplication rate, 1 minus duplication rate. MapQ, we already gathered ourselves. IDX stats, we gathered ourselves. So all these metrics we can do ourselves, probably a little quicker than in the AttackSeq QC. These two, though, we want to be able to get. So to extract them from our AttackQC object, because it's a list, it's going to be super easy. I can just do double square brackets and put this in the quote, or I can do $ and then that slot, and it will give me this metric. Okay. So just to talk you through what PCR bottleneck's about, really is it's aimed to identify PCR bias, overamplification, which can happen with some of these samples. And it's calculated as the number of positions in the genome with exactly one read mapping uniquely compared to the number of positions with at least one read. So if we have 20 reads, 16 map uniquely to locations, 4 do not map uniquely. Instead there are 2 locations, so they map to 2 locations, both of which two have 2 reads will lead us to a calculation of 16 over 18. A PBC of 0.889. What we really want to know is what is a good score here. And ENCODE, this is the great thing about these big consortiums, because they run hundreds of data sets, they can kind of set top and bottom boundaries. And they would say 0.7 would indicate severe bottlenecking, 0.7 to 0.9, moderate, 0.9 and above, no bottleneck. Good. No, you'll just say it's acceptable at that level. So there's not much cleaning up you can do. I mean, people have tried methods for like removing duplicate reads. The issue with duplicate reads, and me and my old boss wrote a paper on this, which is still contentious now, but I think generally accepted, is that once you get enough sequencing depth, you start to get duplicates which are real. So removing duplicates can be a really dangerous thing. And if you can imagine you have a 36 base pair read on each strand, it means the maximum height you could have before you're a duplicate is 72. And it's that limiting the maximum range of your signal became like a real problem for differential AttackSeq and differential ChIP-seq. You just have a max, and then another one's still the max, but it's actually way higher since we removed duplicates. So there's not much you can do to clean it up, but if you had a sample which fell below this, I would start to think about maybe rerunning that sample. Most of the time, you're going to be, I mean, this is ENCODE data. You can see it's very close. most of the time with your data nowadays, you'll be quite high, quite a lot higher. And without slagging ENCODE, ENCODE got there first. So they were doing these methods when they were still developing. So a lot of their quality metrics are quite low. PCR bottleneck 2 is a secondary measure, and it's calculated very similarly. So rather than, so it's the number of reads which map exactly one position uniquely compared to the reads which map exactly two positions uniquely. So not two or more, two positions. So then we could worry here that values less than 1 would be really severe bottlenecking. 1 to 3, moderate, 3 and above, no bottlenecking. As we can see here, we extract it the same way that we extract all the others. $, the element we're retrieving. And then we can see we beat our score there as well. Right. So this is really, it's a way PCR bottleneck of looking at the complexity of your data, library complexity. That's the end of this session. I'm going to have some exercises here, and they do start quite a low level, but you do end up creating an actual bigwig of the different types of signal. What is kind of cool here, as you can see, I mean, we'll come to it, but I'm not sure how much you know about it. You can kind of see the nucleosomes piling up either side of the TSS, which is often the case. You'll have like a -1 and +1 nucleosome. Also, I think in this one, we're also going to use some of Christina Leslie's data, which is kind of interesting because Christina Leslie's data is so much cleaner than the ENCODE data, but it is years on. What you can also see, though, is that Christina, as you get to it, she didn't have any fragment lengths longer than a certain range. So she must have cleaned up her data at some point. So to get rid of these ones we saw in the Greenleaf paper where they had, 1, 2, 3, 4, 5, 6, 7, 8, 9. You don't get that in modern day ATAC-seq. It's only the first 3. And I guess they do some size selection and clean it up quite well. You can actually see that for real in her data. Okay, so I'll leave you guys to this. I'm going to put all the videos up, so I'm going to try and do that right now. But I'll speak to everyone next week. No, Friday this week. Monday today. Sorry, cancel. If you have any questions, just let me know in between. I'm always here, available.